New Test Method Validation/Verification by Tariq Mohammed Hammam. Validation versus verification. The terms validation and verification are used to identify the processes employed before implementing a new test method in the clinical laboratory. Some laboratory professionals use the terms validation and verification interchangeably or just use the term validation to refer to both processes, but to clarify. For tests that have been cleared/approved by the U.S. Food and Drug Administration (FDA), but have been modified, or for laboratory-developed tests (LDTs), the laboratory must validate. In these two cases, the laboratory is acting as a manufacturer and must establish performance specifications of a test method. Validation includes the following, at a minimum: 1. Accuracy. 2. Precision. 3. Reportable range/analytical measurement range, AMR, for quantitative procedures. For reference intervals, RI. 5. Analytical sensitivity, lower detection limit. 6. Analytical specificity. Other studies as applicable. 1. Test interferences. 2. Carryover studies/cross-contamination. 3. Interferences. 4. Matrix effects. 5. Analyte stability, reagent stability, specimen stability/specimen types. 6. Linearity studies. 7. Determination of controls. Need to devise an individualized quality control program, IQCP, calibration procedures. When a clinical laboratory desires to implement a new method or test system and analytes that have been cleared/approved by the FDA and that the laboratory has not modified from the manufacturer's instructions, that laboratory is obligated to verify manufacturer claims. At a minimum, verification includes the following analytical characteristics: accuracy, precision, reportable range/analytical measurement range, AMR, for quantitative procedures. Note, verification of the reference interval, RI, is also required. Verify that the manufacturer's reference intervals are appropriate for the laboratory's patient population. In addition, the use of IQCP, in place of CLIA/QC requirements, may be evaluated at this time. Experiments the laboratory should conduct to verify performance characteristics include: 1. Method comparison to verify accuracy. 2. In-run repeatability and within laboratory precision that is performed over a period of time to verify the precision. 3. Linearity studies and/or analytical measurement range, AMR, verification to verify the reportable range. The Clinical Laboratory Improvement Amendments, CLIA, dictate that every clinical laboratory needs to evaluate every new non-waived method before placing that method into use. Evaluation for a qualitative method. One evaluation of a qualitative method usually involves a comparison between the new method and an established method to determine if the same results are obtained using both methods. The problem with the comparison between qualitative methods is that it may be difficult to determine which result is correct and which is not correct. Any discordant samples should be repeated by both methods for confirmation. A fourfold table can be constructed to compare the two methods. The table, as shown on the right, contains two variables, the new method and the established method, each with two levels of variables, positive and negative. The calculation shown below the table can be used to evaluate the correlation. Acceptable correlation is often considered to be greater than 0.95, or 95%. In the example shown below the calculation formula, the measurement of agreement appears to be satisfactory. The two discrepant results in the example may be attributed to chance variation between methods. The two discrepant results could also be explained, for example, by a difference in test sensitivity. Reviewing the manufacturer's package inserts may reveal differences between test sensitivity and specificity. For a qualitative procedure, sensitivity is not the detection limit, as it is with quantitative tests. Rather, it is the point at which a test is considered positive, the cutoff. The specificity of the test is the ability of the method to accurately report a negative value when the test is indeed negative, below the cutoff. Evaluation using previously tested proficiency testing samples with known results for the method being tested may also provide method verification data. In this case, it is important to ensure that the samples were properly stored and are at room temperature when testing commences. It may also be possible to evaluate the new method against clinical symptoms and/or supplementary tests. If a patient is diagnosed with the disease or condition based on clinical symptoms and/or other types of tests, and the result of the qualitative test that is being verified is positive, this information can be documented as part of the method verification data. For example, a rapid strep A method is being verified and a patient test result is positive using the rapid method. A strep culture is set up simultaneously and the result of the culture is positive for beta hemolytic group A streptococcus. Therefore, the rapid qualitative method is accurate in this case. This information should be included in the method verification documentation. Method validation for specimens other than those approved by the test method manufacturer. The methods used to perform testing on body fluids and other specimen types also need to be evaluated. The method performance specifications that apply to blood specimens may also be used for body fluids if the manufacturer states that the method procedure has been tested for fluids other than blood, e.g., urine, cerebrospinal fluid, synovial fluid, and no matrix interferences exist. If the test method has not been evaluated by the manufacturer for matrix-related bias in other body fluids, published literature citations must be included in the procedure manual, or laboratory personnel will need to perform their own testing and document the absence of matrix-related bias. The laboratory may use one of these suggested evaluation methods, although other methods may be equally valid. 1A dilution study using admixtures, a product of combining two or more substances, of samples. 2 spiking samples. Determine when a complete method validation is necessary and when performance verification is sufficient prior to implementing a new test method. Spiking studies can be conducted using calibrators, controls, or serum, provided the volume change is less than 10%. When choosing a diluent, options include manufacturer-recommended diluent or a matrix-similar choice such as a 7% albumin solution. In any case, the most important criteria for determining the extent of method performance validation is clinical relevance. The laboratory's medical director should be involved in determining what needs to be done to validate the test method for body fluids and other specimen types to ensure patient safety. For fluids and other specimen types that are not routinely tested, it may not be possible to establish test performance characteristics to the same extent as blood specimens, for example, analytical accuracy and reference range. If this is the case, but physicians request this testing because they find clinical value in it, a note should be included in the patient report stating, for example, the reference intervals and other method performance specifications have not been established for this body fluid. The test result must be integrated into the clinical context for interpretation. Other situations when method verification is required. Accuracy, precision, and reportable range (AMR) must be verified in these situations. 1. An instrument is moved. Verification of method performance specifications ensures that the system was not affected by the relocation process or any changes due to the new environment, such as temperature, humidity, or reagent storage conditions. 2. Multiple instruments of the same make and model are purchased. Method verification must be completed on each instrument. 3. A test is reintroduced into the laboratory's test menu. Tests may be reintroduced into a laboratory's test menu for several reasons. For example, the testing is only needed during certain times of the year, intermittent or seasonal, e.g., influenza antigen testing. Laboratory management decides to bring a previously discontinued procedure back in-house, or the instrument that was used for that test had to be sent out for repair and is now back in use. When the test is reintroduced, performance specifications must be verified again, within 30 days, prior to patient testing. In addition, proficiency testing must be performed within 30 days, and competency for analytes must be performed within 12 months prior to restarting the patient testing. For a new analyte is added to an existing test system. Verification of the new analyte method on this existing test system must be performed. Note, verification of the manufacturer's reference intervals, RI, is also a part of the process. Primary responsibilities related to method validation/verification. The persons responsible for a method validation/verification need to 1. Apply a clinical perspective to the task. Decisions about whether to accept a new method should be based on the clinical impact of the method. 2. Set a target, a performance standard, before beginning. This analytical goal is related to the first bullet point above because clinical criteria are the best sources of these allowable errors. 3. Perform experiments that gather representative data about the analytical performance of the method. 4. Use statistics wisely to convert the data into estimates of error. 5. Compare the error estimate to the allowable error to make an objective decision about whether to accept the method being evaluated. Quiz. Question 1. Physicians want the laboratory to start performing amylase body fluid testing. The manufacturer makes no claim regarding any fluid testing other than blood. What steps, if any, are necessary before the laboratory begins testing body fluid for amylase? Please select the best answer. 1. The laboratory must test for interferences only. 2. No further testing is needed. If a test method has been evaluated by the manufacturer for blood, it is also acceptable for testing body fluids. 3. The laboratory must validate the reference range only. 4. The laboratory must perform matrix-related bias testing for amylase on body fluid. The correct answer is the laboratory must perform matrix-related bias testing for amylase on body fluid. In this case, the laboratory must test and document the absence of matrix-related bias for amylase on body fluid. Additional testing is indeed required. The medical director has to be involved in the method performance validation. It is not always possible to test for interferences or establish a reference range for a body fluid. The medical director would have to research published data in order to make an informed decision. Question 2. What are some ways a laboratory could test and document the absence of matrix-related bias in body fluids? Please select the best answer. 1. Running different levels of QC material. 2. Running linearity samples. 3. Using samples that have not been spiked. 4. Performing a dilution study using admixtures of samples. The correct answer is performing a dilution study using an admixture of samples. A laboratory can also evaluate matrix-related bias in body fluids by spiking samples. Linearity material is used to validate the analytical measurement range and is not the correct answer. QC is used to determine if a test system is performing correctly, not for evaluating matrix-related bias. Question 3. Which of the following processes should a laboratory include when validating a new test system versus verifying a new test system? Please select the best answer. 1. Reportable range. 2. Accuracy. 3. Precision. 4. Analytical sensitivity. The correct answer is analytical sensitivity. A laboratory must validate a test system that has been approved by the FDA but has been modified by the laboratory or any laboratory-developed tests, LDTs. A laboratory must verify a test system that has been approved by the FDA and has not been modified by the laboratory. Testing for analytical sensitivity, analytical specificity, and interferences are examples of several additional items necessary to complete when validating a new test system. Reportable range, accuracy, and precision are involved in the validation, as well as in the verification of a new test system. Question 4. A laboratory is in the process of changing to a different method for analyzing urine amphetamines. Which tool can the laboratory use to compare qualitative amphetamine results from two different methods? Please select the best answer. 1. Linearity material. 2. Twofold table. 3. QC material. For using previously tested proficiency samples. The correct answer is using previously tested proficiency samples. A fourfold table, not a twofold table, can be constructed to compare two qualitative methods with two different variables. Also, testing previously run proficiency testing samples with known results for the testing method can be used. Linearity and QC material cannot be used to compare a test method. Thank you for watching. Like, share and subscribe to see next video.